Good afternoon, everybody. Welcome and thank you for joining us. It is my great pleasure to welcome you and to introduce today's speaker, Professor Kristi Kiick, this year's speaker for the Duncan Dowson Memorial Lecture. My name is Alvaro Mata. I'm from the University of Nottingham. And before I introduce Kristi, I wanted to give a bit of background to the area of biomaterials, to tell you how we got to where we are today, and why this is such an important area, and to get an understanding of why the opportunities are so exciting. So, if we go back in time, we can find biomaterials being used in medical applications for tens of thousands of years. Most of these materials, or all of these materials, were used up to the 1920s, were found in nature. These were natural materials. And you can see here's a picture of a prosthetic eye from thousands of years ago made out of mud and wood. So this is the initial type of materials that were used as biomaterials. Then between the 1930s and 60s, we find materials that were not designed for medical applications, but were designed for other things. And as you can see in these dates, a lot of them were designed for war. For example, if you see polymethyl methacrylate was a material that was used to build windows in warplanes. And when they were hit, the pilots, when they would go to the hospital, they would see pieces of this PMMA in their eyes. And they saw that they were compatible. They were inert. There was no reaction to them. So that's how they found that these were, they could be used as biomaterials. So this is an example of how these materials were designed during these times, how biomaterials were discovered and designed. Then from around 1960s, we began to have finally materials that were specifically designed for medical applications. And here we started to develop materials and to see materials that were with the concepts of biocompatibility. Can they be compatible with the human body to avoid rejection, for example, or can they degrade in time controllably? These were concepts that were beginning to be designed in these materials, again, from around 1960s, and these concepts really continue to today's designs. And finally, from about 2 decades ago, we began to have the capabilities to design materials from the molecular scale, molecularly designed materials that now we could begin to implement with new properties. Can we make them smart or intelligent, or can we have materials that can recreate properties of living systems? And this is quite an important concept with tremendous opportunities. Kristi is one of the world leaders in this area. And just to finalize this introduction, I wanted to show you why this is so important. And to do that, we need to remember that nature in biology, in our bodies, we are all hierarchical. We begin from the atomic scale and acquire order at different scales up to amino acids, proteins, cells, tissues, and organs. And perhaps the most important, or at least the most functional aspect or part of this hierarchy are proteins. Proteins are really what do things in our bodies. And as you can see in this slide, there are a number of functionalities that they do. So you can imagine if we were able to harness these properties and harness proteins to create materials, you can imagine the types of materials that we could end up to have tremendous functionality. And so this is what brings me now to Kristi. And you will see what Kristi's design are. But just to give a quick introduction to Kristi, she is Blue and Gold Distinguished Professor of Material Sciences and Biomedical Engineering at the University of Delaware. She's a fellow of the National Academy of Inventors, American Chemical Society, American Institute for Biological and Medical Engineering. She has published more than 150 articles, books, or patents, has numerous awards, as you can see here in this slide, and is also a member of a variety of advisory boards and editorial boards. So it is a great pleasure an honor to have Kristi with us today. And Kristi, the floor is yours.
Thank you, Alvaro, and thank you to the Institution of Mechanical Engineers for inviting me to give this Duncan Dowson Memorial Lecture. You'll see, although my work is related largely to the design of materials, both at my title in the published arena was in the nanostructure, but we also do microstructured polypeptide materials, is aimed at applications that mirror some of the type of work in tribology that Professor Dowson worked on. So our goal is to help people not need the implants where the biomechanical tribology questions come into play. We're hoping to address some of those health challenges upstream. Before I get started, I wanted to give a brief introduction to where I'm from. As Alvaro mentioned, I'm in the Department of Material Science and Engineering and also in Biomedical Engineering at the University of Delaware. Many people don't know that Delaware is even a state. Many people in the U.S. sometimes don't even know that, despite the fact that Delaware was the first state in the U.S. Is the first state constitution in the country. The University of Delaware, where I have started and maintained my career, is one of the oldest institutions of higher education in the U.S. It was established in the mid-1700s, only shortly after the University of Pennsylvania. So there's a long history of academic and research excellence in the region and on our campus. Our campus is a beautiful place to study and work. I had the very good fortune when I started as an assistant professor to be housed in a brand new building, which you can see on the bottom right of the slide, which is named DuPont Hall because of the DuPont family's long history of philanthropy and scientific excellence in the region. My group at the University of Delaware is interested, as Alvaro mentioned, in designing polypeptide or protein-based materials, you can say, for tissue regeneration. There's many places in the body where these materials can find use. We have applications both in multiple arenas. In the first, we make injectable microstructured hydrogels that are used for applications in healing tissue after cardiovascular surgeries. We have new projects that are developed based on my sabbatical work, my Fulbright scholarship work at the University of Nottingham this past year, where we're now taking some of our polypeptide materials and we're looking at manufacturing them in ways that we can make high throughput scalable methods for making microarrays of stem cells. And then as we progress into the lower regions of the body, we're interested in developing some of our nanoscale carriers where we're using the ability of materials that are functionalized with collagen as a way to target collagen tissues so that we can design nanomaterials that can carry drugs to treat osteoarthritis and other arthroses, as well as designing collagen functionalized carriers that are motivated and inspired by the extracellular matrix as a way to address the poor healing of chronic wounds. For the purposes of the talk today, I've broken our research into two short vignettes. I will talk first about the injectable microstructured hydrogels and our motivation for doing that work. And then we'll have a short break to answer questions, and then I'll present another short vignette on collagen targeting peptide-based drug carriers. when one is interested in making hydrogels that can help heal tissue in vivo, there are three main components to consider. We have, first of all, importantly, the scaffolds, the interaction of cells with that scaffold, and the manner in which the engineering of the scaffold can control what the cells can do once they grow in the scaffold. And another feature of that is the incorporation of therapeutics. We have spent a lot of time in my group thinking about how we can design scaffolds. And in the past 10 years or so, we've been very interested in the use of proteins in vivo that have elastomeric behavior. And we've been interested in understanding how those materials can be produced in microstructured formats, and in this particular application, for cardiovascular applications. So at this point, I would like to ask the audience a few questions about cardiovascular conditions. So I have three poll questions. I think the first says it's already been triggered. There is, okay. I'm navigating this on the fly here during the talk, so if it should be appearing on your screen now. How many people a million people in the UK suffer from cardiovascular or circulatory diseases, which is abbreviated CVD? So please just give your quick knee-jerk reaction to what that number might be. I'm going to gather the poll results now. So I'll close the poll in 15 seconds. 10, 9, 5, 3, 2, 1. Let's publish the results. Okay. So how many people? 10 million. So 42% of the participants who responded indicate 10 million people are affected by CVD in the UK. That number is actually not the numbers that are reported most recently by the British Heart Foundation. So that's good news, because the number that's reported is that 7.5 million people in the UK suffer from cardiovascular disease. We can contrast that. I'm posting the second poll to what is, just 1 second, please. Okay, there we go. The second poll question, which is how many million people in the US suffer from cardiovascular disease? So the numbers are much larger, 15, 50, 85, or 120 million people. Go ahead and get a quick response to that. Leave the poll open for 15 seconds. We have 10 seconds. 3, 2, 1. So we'll go ahead and gather the poll results. This one, 85 million. That is exactly, well, yes, very close. The actual number is 82.5 million. So as you can see, cardiovascular disease is an enormous problem in the United States. So there's lots of motivation, not only in the U.S. but worldwide, to solve problems with cardiovascular disease. So for the the poll 3 is about osteoarthritis. Please don't answer that one just yet. I thought I had a third question about cardiovascular, so apologize for that small error there. So cardiovascular disease is a huge challenge in our two countries and also worldwide. Cardiovascular disease affects 82.5 million people in the U.S. now, but is projected in just 15 years from now to affect over 130 million adults in the U.S. The health care costs of that are enormous, and it's a very huge problem. The surgical intervention for helping to treat patients who suffer from cardiovascular disease is coronary artery bypass grafting surgery. Those surgeries actually have high failure rates, and I'm here to tell you a couple of reasons why. So CABG, as people refer to it in my circles, is the process by which a vein harvested from elsewhere in the body, usually the saphenous vein in the leg, or an artificial vein material that is constructed with synthetic polymers, such as the polyesters, is used to replumb the heart. So an area that's shown by the inset in this slide where there's decreased circulation in an area of the heart owing to atherosclerosis or some other restenosis or closing of the blood vessels is addressed by taking another conduit and circumventing the blood circulation around that blockage. There are two main surgical procedures that are used for this type of surgery. One that's called skeletonized, and the other is use of pedicled vessels. So the skeletonized vessels so a lot of what is chosen depends on the surgeon's preference, but skeletonized vessels are ones in which the tissue that directly surrounds the vessel is removed. It's been shown by analysis of clinical outcomes that skeletonization of a vein allows a surgeon to work more effectively with longer grafts, so it gives them more surgical flexibility. And there's also been shown to be a decrease in the number of infections post-surgery and a decrease in the amount of time it takes for recovery. So as you can imagine, if you're skeletonizing a vessel, you're manipulating in as uninvasive a way possible, but still very invasive, the exterior portion of the vessel. So our idea is whether or not we can place injectable hydrogels and just use them to bathe the exterior portion of a skeletonized blood vessel after the tissue has been removed and just after it's been sewn into circulation after the surgery. The hydrogels have to adopt very specific mechanical properties and behaviors. What I show in the bottom right of the slide here is a sort of a side image of a blood vessel showing the multiple layers that comprise a blood vessel. The most interior layer is populated by endothelial cells, and those are the cells that mediate the interface of blood and blood flow through the interior lumen of the blood vessel. In the middle layer, the media, there are smooth muscle cells that are responsible for controlling the contractile actions of the blood vessels to help move blood through circulation. And then on the exterior part of the blood vessel, what's shown here is sort of like this little pink spaghetti-like layer is called the adventitia. That layer is populated by two different cell types, fibroblasts, which are a primary cell component of tissue, and then a special type of endothelial cells that help control the formation of a microstructured vascular network that nourishes the cells. So our interest is in making hydrogels that can have excellent physical properties, that can be administered simply, have good mechanical support, can be elastic, and in this case, also elastomeric, and that can support the diffusion of nutrients into the material as well as waste products out of the material from cell action. These materials also need to control, as I mentioned, the main activities of cells, their proliferation, migration, and in the case of cardiovascular applications, the generation of blood vessels. We seek to do this by adding a secondary component that allows the gels to be microstructured. These domains, or the second phase that we introduce into the materials, is intended to enable the localization not just of micromechanical properties in the material, but to allow us to control where cells can migrate and proliferate in these materials, and also to serve as delivery depots. Our particular approach to making these microstructured hydrogels takes advantage of the liquid-liquid phase separation that different polymers and proteins can undergo. What I show on the left is a simple rendering of the coexistence curve for a phase separation process in which a single phase is observed if you're below the parabolic shape. But once inside the envelope traced by the curve, phase separation happens. Phase separation can happen by two different mechanisms. One is indicated by the binodal curve, which is a nucleation and growth mechanism that results in the formation of small circular domains that mature and grow in size over time until, as you progress to the bottom layer, one observes a macro phase separated solution, much like you would see if you left an oil and water solution or emulsion sit over time. If one is able to capture phase separation on the spinodal line, a different mechanism occurs, and in that mechanism there is a bicontinuous set of two phases generated that then matures into a sort of, you can imagine it looking like a vascular network, that one side of the gel would be continuously connected through a single phase to the other side of the gel. And if one can capture that phase separation in the earlier stages, you can capture this vascular-like network. This phase separation process, sometimes you'll see it referred to as ELP-CLPs on my slides, is very tunable. So there's a wide variety of parameters, including temperature, pH, molecular weight of the materials, and their concentration that can be used to control the thermodynamics and the kinetics of this phase separation process. Both natural and synthetic polymers can undergo these types of processes, and both, as Alvaro nicely introduced at the beginning, both are relevant to the design of biomaterials. Natural polymers, in my group we use protein-based polypeptide materials, but also things like polysaccharides also have benefits to these applications. They are cell interactive and biodegradable, but the natural polymers do suffer cons of batch-to-batch variability, and it's difficult to tune the materials properties of these natural polymers because they've been evolved to have a set of properties to fulfill their function in nature. Synthetic polymers, on the other hand, are very reproducible. There's lots of tunability in the versatility of chemical synthetic methods to make them, but they have limited bioactivity and can have potentially harmful degradation products once introduced in the body. So we try to take control of the advantages of both classes of these polymeric materials to make modular, tunable, cell interactive, and biodegradable protein-based polymeric materials. Our inspiration for these materials comes from the structural proteins found in nature. These materials have their outstanding mechanical and biological properties because of the exact control of their monomer sequence in the chain. So if we look at the resilin region of this graph and look at the amino acid designation, so these single letters, GG, RPS, that is, each letter represents an amino acid that confers to this chain specific chemical and structural properties. So because of that exquisite level of control, one can control the conformations or the structure of the chains on a local level, as shown for the resilins. And as you progress through this set of structural materials, you can see higher orders of structural control as you would go through the elastins and then the silks. And then once one considers the collagens, the keratins, and the B silks, individual chains will then hierarchically assemble into larger order structures that then can carry significant load. So we've looked a lot at resilin, elastin, and collagens, and I'll tell you a little bit more about why. So some of these protein-based polymers can exhibit, as I mentioned before, very rich phase behavior. So looking at the phase separation graph on the left, the green shaded area indicates the one-phase regions where everything is soluble in solution. If one progresses out of this green shaded area to lower temperatures, so down in this graph, the one traverses the upper critical solution temperature, or the UCST. That phase transition is controlled by a range of dipolar type interactions in the polypeptide chain. And because we can engineer and specify where specific chemical groups are located on the chain, that gives us a great deal of flexibility in controlling the temperature at which this phase separation happens. If we go back to the one-phase region and instead increase temperature, one traverses the lower critical solution temperature, in which phase separation occurs as a generally hydrophobic molecule becomes less solvated and then forms a polymer-rich phase inside a solvent-rich phase. So we can take advantage of these individual side chain interactions on our protein, as well as overall hydrophobicity, as a way to tune these phase separation processes. And we have done that largely by controlling the amino acid sequence. That allows us to use a specific set of conditions each time we form a phase-separated architecture, and to do that under conditions that are compatible with cells. So while it may seem really difficult to do the engineering, it's really not that hard. We have a great deal of versatility in the molecules we make. The molecule that we're using for this particular purposes of this talk are resilin-like polypeptides. Natural resilin is a highly elastomeric protein. It's found in organs of insects and is involved in very efficient energy storage. If you load these materials and then unload them, there's essentially zero hysteretic loss in the stress strain curve. Because of this, these materials can undergo very high frequency movements at very high numbers of cycles. And even over the short lifetime of an insect, that's important because wings vibrate at, and sound-producing organs vibrate at extremely high frequencies. The resilin is naturally fluorescent, as is shown in the bottom image from the tendon of a dragonfly, and it allows us to easily identify where our resilins are located. REC1 resilin from fruit fly, the gene sequence for encoding that material has been known now for 15 years, and it allows us to make mimetic materials where we can control and change the amino acid sequences slightly to make resilin-like polypeptides that mirror the natural resilin properties. So we can make materials that are low stiffness, high extensibility, and very efficient energy storage. We make our materials, they undergo both, because of the sequence control that we have of these materials, they can undergo both the upper critical solution temperature transition and the lower critical solution temperature transition as well. We make these genetically, which allows us to incorporate not only the structural features of the RLP, as is shown in a very simple schematic on the far left of this slide, but also incorporate different bioactive domains. So we can incorporate cell binding domains, as shown in green, heparin binding domains, as shown in purple, and then MMP, or matrix metalloproteinase, so enzyme degradable domains as well, so that these materials can be degraded once they're deployed in vivo. We make them in bacteria. As I mentioned, the DNA for encoding the sequence of the resilin-like polypeptide is known. We can design and order these days the recombinant plasmids, so just circular pieces of DNA that we then incorporate in multiple copies. That's not shown here, but in multiple copies in an individual E. coli cell. Those E. coli cultures are then grown up in flasks or high cell density fermenters, and those cells are then directed to make our protein. When we design our protein, as is shown in the bottom right slide, the polypeptides are encoded with a tag shown in green that enables their rapid and easy purification. So then we can just lyse the cells open, take all of the interior components of the cell in solution, but then easily purify them so that we can end up with a pure material that is just our protein. We can easily confirm the purity and the composition of our materials by using analytical procedures that are quite common in the biochemical and protein-based materials community. Gel electrophoresis simply separates the protein polymers on the basis of their molecular weight. What's shown on the left is a ladder of a variety of different proteins that have different known molecular weights. And then, as you can see, we can inject into the gel our RLP products, and they are very pure, and they electrophorese at a molecular mass that is consistent with what we expect from theory. So these polypeptides have molecular masses on the order of 25,000 grams per mole. And when we do MALDI mass spec of these materials, as you can see, we get a single mass to charge peak for the RLPs that is consistent with its theoretically expected mass. So mass spectra don't look any better than this. If you use a synthetic polymer, even the ones under the most highly controlled synthetic conditions, one will observe an envelope of different molecular weight species in their sample. So these materials are pure, they're a singular molecular weight, and we know by amino acid analysis that their composition is exactly what we expect. Because we've engineered the sequences to carry chemically reactive groups, in this case amine groups, the NH2s, we can functionalize the RLPs with reactive groups, shown here these double bonds on the far end of the linker after it's connected to the RLP, that allows these materials to be photo cross-linked. So we can make these photo cross-linkable materials. They're functionalized with acrylate groups, so the same sorts of monomers that are in the acrylate and methacrylate residues, polymers that Varo introduced earlier in the talk, with the addition of a photo initiator and then irradiation with UV light. We can make hydrogels that have spatio-temporal control. These materials, as I mentioned earlier, have good and tunable mechanical properties. As shown here in the bar graph, you can see just by changing the concentration of the materials, we can get shear storage moduli that are perfectly consistent with what we expect in soft tissues, and that the RLPs are not toxic to cells. You can see there's good cell viability, and in fact really high cell viability in these materials, but only when they carry the RGD domain, which is a canonical cell binding domain, do cells spread on these materials. And I show here tensile loading and unloading of over 10 cycles of a resilin-based material, illustrating the great resilience and the lack of hysteresis in the loading and unloading of these materials. So when we want to induce phase separation, what we do is make a solution of the RLPs, warm the solution up so it's in the one phase region of the phase separation graph, or the phase separated diagram, and then we can cool the solution below the UCST, introducing a phase separation. We've done that by easily incubating these materials on ice, but you can also incorporate it in a plate reader or some other form of temperature control. The gels are then illuminated by UV light, and then the gels can be easily collected from these synthetic protocols to make microstructured gels. We know that the microstructured gels can be made to have open domains that are on the length scale relevant for cells. The scale bar in these microscopy images, these are confocal microscopy images where the light color is the RLP, so we can pick the autofluorescence up of the Resilin-like domain. At low concentrations, the phase separation is slower, so the domain sizes are much smaller. When we increase the polymer concentration of the solutions, the polymers can phase separate more rapidly, so at a given point in time, we're able to capture a size domains on a size scale that are more commensurate with that of cells, which is on the order of tens to hundreds of microns. What's important for us is that when we change the sequence of the RLPs then, we have a very easy way to tune the length scale of the dimensions of these RLP phases, again, shown in the light color across hydrogels with different structures. So I'm not at liberty yet to reveal what these sequences are, but suffice it to say that we're able to control not only the kinetics of the phase separation, the cell binding properties as well. And we've shown this in related work where we incubate RLPs that are photo cross-linkable. In this case, they're also combined with polyethylene glycol, so a PEG acrylate. That's for different reasons, but they're photo cross-linked. The microstructured hydrogel still forms. And what you can see is the blue is the autofluorescence of the RLP domain. So you can see that they're regularly distributed throughout the matrix. They're very, very cell-friendly. So the green images are the calcine dye that stains live cells. We also have in here a red dye, Ethidium homodimer, that will stain the cells red. And you can see that there are no red cells really to be seen in these materials. And then the merged image on the bottom right of each panel shows that the cells are isolated in the regions where there are RLPs. So we're really excited about deploying these materials in surgical venues with cardiovascular surgeries. We're in the process of doing those studies now. What I'd like to have been able to show you in this first portion of the talk is that these protein engineered polymers that we work with are highly tunable, very biodegradable, bioactive and biodegradable. We can make materials with exact structure and sequence. That allows us to control with fine detail their thermally dictated liquid-liquid phase separation, and that we can tune the microstructures by using these types of approaches. So I would like to pause at this point of the presentation to see if anyone has any questions that they'd like to ask about the Resilin-like polypeptides. Alvaro, are you able to monitor?
Yes, Kristi, just getting there. No, I have a question by Michael Howell, who asks, what is the success rate of CABG, coronary artery bypass grafting?
Yeah, thank you. That's a great question. The current long-term patency rate, so the period over which the grafts remain open at 5 years, is still only on the order of 50%, particularly for the smaller diameter grafts. So there's a real need to improve this patency rate to much higher percentages. There's not only the health reasons to help manage that on behalf of the patient, but that every additional surgery that a patient has to undergo is potentially dangerous, and then also the burden not only to the individual patient but also to the healthcare system. So there's still a really significant need to improve the success rates of these surgeries at the longer term time points.
Okay, Kristi, I have one more question from Joseph Langley. Are there any anticipated applications in the areas of orthopedic or sports medicine?
Yeah, that's a really great question. We have not deployed these materials for those types of applications yet. but I would be delighted to consider them. You will see in the next part of my presentation that we are starting to develop drug delivery carriers to help address some symptoms and disease progression in osteoarthritis, but we haven't looked at deploying the Resilin-like materials in the case of trying to improve outcomes of healing in sports medicine injuries, and that's a really interesting potential area to consider these materials.
And Kristi, we have a couple more questions, but I'm aware of time. We should be finishing the talk in about 10 minutes. Would you like to continue? I don't know what you prefer.
Why don't we take the questions and then I can more quickly go through the second part of the talk.
Okay, perfect. So from Neil Patterson, how long do the hydrogels take to degrade under flow and in vivo condition?
That's a great question. The hydrogels in vivo depends on what concentration of the enzyme degradable domain we include. If we make the materials where there is an enzyme degradable domain in each chain, they degrade very rapidly on the order of a day. When we very lightly modify the hydrogel with the enzyme containing materials, the gels remain, I'm just trying to remember, they remain in place for long periods of time. We haven't done any long-term studies in animals just as yet, but they're perfectly happy in cell culture for periods of months or more, and we just haven't looked out any longer than that just yet.
Okay, and from Tony Barber, are there any potential issues relating to the biodegradability of these materials and their effect on the food chain?
Oh, I haven't considered that question before. I think in the context of the RLP only materials, there's very little concern about the components. We are using naturally occurring amino acids, so the degradation products of these gels are amino acids and short peptides. The acrylation of the materials is not highly dense, so I don't anticipate. We've done some in vivo studies of the RLP materials where we don't see any sort of adverse effect in the animals. So I think at the concentration of the acrylates that we use, that even the degradation products are not likely to have any sort of deleterious effects at the concentrations we use. So I don't anticipate there being any problems in that regard.
Okay, and one more question, maybe a quick answer from Mohammed El-Sawi. Have you ever experienced any post-translational modifications that may affect the RLP properties?
We have not studied that in great detail as of yet. What I can say is that we don't see significant changes in how the materials interface with cells or in vivo when they've been implanted. But I don't know. We haven't done that molecular level of investigation.
Okay. Good, Kristi.
Yeah,
you can continue.
I will continue now with the second part of, and this is a shorter part of my presentation, where instead of focusing on scaffolds, we're interested now in focusing on some of the therapeutics that can be delivered from the scaffolds. And I wanted to get a, in the essence of time, maybe I won't initiate the polls. So I will keep moving on with our applications in osteoarthritis. So we're interested here no longer in Resilin, but we're interested in collagen. Collagen-like peptides are short peptides that have a strong affinity for natural collagen. As you guys may be familiar with, collagen is the most abundant protein in mammals. It has a very repetitive amino acid sequence, that is characterized by this glycine XY motif, where the X and Y positions generally are proline and hydroxyproline, although they can be engineered to be different amino acids if desirable. These short peptide sequences are extremely versatile. You can vary the temperatures at which their folded structures unfold, which is called the melting temperature, or TM, and you can incorporate a wide range of bioactive materials. Lots and lots of people have studied collagen-like peptides because there remains enormous interest in understanding how the peptides can assemble to capture some of the hierarchical assembly and mechanical properties of natural collagen, an electron microscopy image of which is shown on the bottom left. We have had interest in understanding how we can use collagen-like peptides as a way to bind collagen both in vitro and in vivo. Collagen-based materials are used to a great extent in clinical practice as fillers, as wound dressings, as other types of foams and materials employed in vivo. Our idea is that if we can use small collagen-like peptides that have an affinity for collagen, as has been demonstrated for small gold nanoparticles in the image on the right, the top image, or that can be used to isolate dyes, as is shown in these near-IR dyes in the bottom right of the slide, to localize drugs or other compounds in particular places in the musculoskeletal tissue. There are lots of diseases where collagen targeting is relevant and where this targeting is made selective because of the degradation of the collagen. So if the collagen is perfectly healthy and well folded, it's hard for those collagen peptides to actually integrate. I'm going to go back a slide. That can actually integrate into the collagen tissues. So on the bottom right slide, those two images where there's a great intensity of the near-IR dye are in murine models where the skeleton of the mouse is wildly remodeled. So there's lots of collagen breakdown and resynthesis in the skeleton. And as you can see, in those areas of high collagen breakdown, there's a high extent of dye binding and localization. So there's lots of diseases where this is relevant, around bone diseases like osteogenesis imperfecta, but in conditions where we're looking at are largely focused on chronic wounds, which I won't talk about today, and then also osteoarthritis. So this community is probably more familiar with many communities that I speak with about the impact of osteoarthritis on the population. Even now, over 15% of the global population is affected, and nearly 70% of Americans over the age of 55 are affected. Worldwide, 50% of people over the age of 65 are affected by this. It comes at an extreme loss in mobility for people, and then also at annual costs that are staggering. It can be hard to treat osteoarthritis and to arrest disease progression because a lot of the drugs, particularly the small molecule compounds that are known to be efficacious, are cleared really quickly from the knee joint because of the motion and load bearing of the joint. So we're interested in looking whether or not we can localize drug delivery carriers to collagen as a potential to improve care. So we're interested not only in the collagen peptides, which I show in blue on this particular slide, but also the elastin-like peptides. So they're elastomeric like Resilin, but they come from mammalian, they're found in mammalian tissue. And elastins also undergo phase separation, but they undergo phase separation when the temperature is increased. So we are looking at ways to make these sort of vesicle compounds so that at this bilayer shell, where the blue outside part of the bilayers are the collagen triple helices, and then the red part of the layers are the elastin-like domains that want to be shielded from solution at particular temperatures. So if one could make a nanoparticle like this one could consider being able to load that nanoparticle with drugs and to bind it to collagen. And if that nanoparticle can bind to collagen, as is shown in the top right, then if we move to the bottom center of the image, that nanoparticle could either be dissolved by lowering the temperature, if one could apply ice, or by heating the temperature, that collagen triple helix can be unfolded, and then those particles could come off the collagen or be dissolved altogether. So this is a two-component system where we use the collagen-like peptides for targeting tissue and the elastin-like peptides as a way to make these collagen-targeting nanoparticles responsive to temperature. We make the peptides on the solid phase. We add functionalities to the elastin. That's an alkyne group, an azide group to the collagen or the CLP peptides. Don't need to focus too much on this, but these peptides can be made in large quantities. The coupling is a very widely used chemistry called Klick chemistry, and then we're able to make the top right these conjugates that have the thermo-responsive ELP shown in red and a collagen domain that can fold into a triple helix shown in blue. We simply put these peptides in solution, heat them up to unfold the collagen-like domains, and then incubate them at 37 degrees to form particles. And then this is a dynamic light scattering image data showing the particles have diameters on the order of 100 nanometers. They are temperature responsive. They get larger at higher temperatures, and then the particles dissolve at higher temperatures. That happens, the diameters are useful for drug delivery. We know that the particles have the vesicle structure that we thought they might because of Cryo-TEM results that clearly show the sort of bilayer structure on the exterior, on the surface of these spherical nanoparticles. The thickness of that layer can be measured easily in the image analysis, and it's on the order of 20 to 22 nanometers, which is perfectly on par with the expected diameter of a bilayer of these collagen-elastin constructs. We make the vesicles. They're stable. We can make them, and then they're stable to injection if they're injected under conditions where they're stable, but they have a unique temperature responsiveness. At low temperatures, the elastins are soluble, but the collagen triple helix is intact. So what we see are in the microscopy image, in this case just TEM, you can see these very small structures that are aggregates of an undefined sort of these sort of like mushroom-like structures that we have, or the triple helically folded conjugates. If we go from the center microscopy image to the right, you can see that there's a very different structure that we see. It's just like a mess of stuff aggregated on the TEM grid during drying. We know from the light scattering results that those molecules are soluble, and that's because the collagen triple helix unfolds. So instead of having this little mushroom-like structure, we have individually soluble molecules. So that allows us, excuse me, to transition these nanoparticles through different temperatures over repeated numbers of cycles. So you can see in this case, we're traversing through the lower temperature. So we're dissolving the nanoparticles to form the little mushroom-like structures that don't scatter light. So at 0 degrees, these solutions are clear. At 25 degrees for these particular molecules, you can see the inset picture that the solutions are cloudy. These solutions, you can heat and cool them at different temperature ranges and see this type of behavior for a whole host of different molecules we've made and for a wide variety of different temperatures. So the next question.
Kristi, I'm sorry. I have to. Just a couple more minutes, if it's okay.
Yeah, certainly. Thanks, Alvaro. So we can load hydrophobic compounds into this bilayer shown here. I hope you can see it, these little yellow nanoparticles. We can then spin down the vesicles to get rid of any drug or fluorescent compound that's not retained in the vesicles. Then as we move across the schematic on the right, we place the nanoparticles. They're perfectly redispersible into small compartments in solution that are separated from the bath of the solution with a dialysis membrane. And then we can monitor the release of these small molecule drugs over time. If you look at the bottom right, we've done experiments over 3-4 weeks, but I show a 1-week time scale. This is release of dexamethasone, which is a corticosteroid that's commonly employed to address symptoms in osteoarthritis. The drug release of the dexamethasone is linear up until day 7, and when we trigger dissolution of the nanoparticles with temperature, the rest of the drug that's encapsulated is retained. So that gives us opportunities to localize not only dexamethasone, but also other small molecule inhibitors that are in various stages of the clinical pipeline for treating osteoarthritis symptoms or the progression of the disease. We've tested the molecules as well, the particles, to see if they can be retained at the knee joint in murine models. We take mice have small knees, so we have to take a really small volume of fluorescently labeled vesicles, inject them in the synovium of the knee, monitor the fluorescence over time. In this case, it might be hard to see the time points on the bottom left graph, but over a week, and then we looked at the localization and distribution in organs. I'll draw your attention to the bottom central image where the fluorescence of the nanovesicles is monitored in the mouse knee from, I think it's the anterior view. I'm not sure which view this is of the side of the knee. Sorry, I'm not remembering that detail of the experiments just at the minute. You can see that the synovium, the particles are retained. There is a small additional site of fluorescence, which is the injection site. So some of the molecules do get released from the needle. We are just doing these surgeries. We're not quite expert at it yet, but that the nanoparticles are retained for about a week in the joints of these animals. We've done in vitro, situ organ imaging of places where one might expect the nanoparticles to go if they were not retained. So the lungs, the kidneys, the brain, and the liver. And in massively overexposed images on the bottom there, you can see that there's no evidence of distribution of the particles in other organs. So we've just moved labs and we're gearing up to do studies of dexamethasone-loaded particles right now. And hopefully the next time I talk about this with this group, we'll have in vivo data. So that was a really quick summary of some of our collagen-containing nanocarriers. We're able to make particles that are not just spherical like the ones I've shown you, but also ones that are platelet in structure, a wide range of different diameters from 80 to 250 nanometers in the case of the nanoparticles, and then platelets that can have dimensions on the hundreds of nanometer scale. The use of peptides allows us to function to control their structure, so we can get a huge range of sizes, shapes, and thermo-responsive behaviors. We can control the release of drugs, which has been great, and we have other data where we show that we can do that just by changing the identity of the ELP. So we can get very different drug release profiles with different ELPs, and we can localize these carriers in various tissues. So I know that was really quick. The question about using these types of materials in sports medicine, I'm glad I was able to show that information. So as an overall summary, protein-based polymers and peptide-based polymers are really useful materials. We can control their behavior. We can make micro, nano-scaled structures. And we can make materials both with useful mechanical properties and also with useful drug delivery properties. I would be remiss if I didn't thank the very talented students in my lab, and I'm thankful for their hard work and effort, as well as my colleagues, both at UD and at the University of Nottingham. So with that, if there's time for any questions, I'd be happy to answer them. But thank you so much for the opportunity to deliver this lecture today.
Thank you, Kristi. Thank you very much for this wonderful talk. It's wonderful to see this repertoire of smart biology-inspired proteins that you are developing. I think in the interest of time, we're going to have to finish now. I do want to mention a message by the Vice Chair of the Biomedical Engineering Division at the Institution of Mechanical Engineers, Sylvain James, thanking you for your time and this wonderful lecture.
Thank you.
Yes, great. And I want to just thank everybody for joining us today. Please stay safe, and we look forward to seeing you next year. So thank you, and thank you, Kristi. Bye-bye.
Thank you so much. Bye-bye.